Hello everyone, welcome to my channel Expert Valley. So this video is all about PCR, Polymerase Chain Reaction. Introduction of PCR. PCR or Polymerase Chain Reaction is a technique used in molecular biology to create several copies of a DNA segment. This technique was developed in 1983 by Carey Mullis, an American biochemist. It is rapid and in vitro method for amplifying defined target sequence present within the source of DNA. This method is designed for selective amplification of the target DNA sequence. PCR has made it possible to generate millions of copies of a small segment of DNA. This tool is commonly used in the molecular biology and biotechnology labs. Principle of PCR. Polymerase chain reaction is the technique used for the amplification of the defined targeted DNA based on the principle of enzymatic replication of the nucleic acid. Primer, a short DNA segment, is added for the further polymerization. DNA polymerase synthesizes new strand of DNA complementary to the template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3-hydroxyl group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. Components of PCR. Following are the components: DNA template, DNA polymerase, oligonucleotide primers, deoxyribonucleotide triphosphate, buffer system. DNA template. The DNA of interest selected for the amplification and for the generation of copies. DNA polymerase. Tag polymerase is used. It is thermostable and does not denature at very high temperature. Oligonucleotide primers. These are the short single-stranded DNA complementary to the 3' end of sense and antisense strands. Deoxyribonucleotide triphosphate. These provide energy for the polymerization and are the building blocks for the synthesis of DNA. These are single units of bases. Buffer system. Magnesium and potassium provide optimum condition for DNA denaturation and renaturation. It is also important for fidelity, polymerase activity, and stability. Primer design. Length of primer should be 15 to 25 nucleotide long with base component of GC about 40 to 60%. It allows selective amplification. Some prior DNA sequence is needed for the construction of primers. Two primers are designed forward and reverse. These primers are called amplimers. Here, reverse primer is for plus strand and forward is for minus strand. Steps of PCR. Denaturation, annealing and elongation. First we'll see about denaturation. Denaturation occurs when the reaction mixture is heated to 94 degrees Celsius for about 2 minutes. This breaks the hydrogen bond between the two strands of DNA and converts it into a single-stranded DNA. The single-stranded now act as a template for the production of new strands of DNA. The temperature should be provided for a longer time to ensure the separation of the two strands. Here in this slide you can see the image how the DNA gets denatured. Now the second step is annealing. Annealing of primer. Here the reaction temperature is lowered to 54 to 60 degrees Celsius for around 20 to 40 seconds. The primer binds to their complementary sequences on the template DNA. Primers are single-stranded sequences of DNA or RNA around 20 to 30 base pair in length. They serve as the starting point for the synthesis of DNA. The two separated strands run in the opposite direction and consequently there are two primers, a forward primer and a reverse primer. Down here you can see the image. Now elongation. This is the third step of PCR. It is also called as extension. At this step, the temperature is raised to 70 to 80 degrees Celsius. The bases are added to the 3' end of the primer by the tag polymerase enzyme. This elongates the DNA in the 5' to 3' direction. The DNA polymerase adds about 1000 base pairs per minute under optimum condition. Tag polymerase can tolerate high temperature. It attaches to the primer and adds the DNA bases to the single-stranded. As a result, a double-stranded DNA molecule is obtained. These three steps are repeated 20 to 40 times in order to obtain a number of sequences of DNA of interest in a very short time period. Here in this slide, it is shown the overall picture of the PCR. All the three steps are displayed: denaturation, annealing and extension. Step number 1 showing denaturation, where the two strands are getting separated at very high temperature. The second step showing annealing, where the primer is getting added, and the third step is extension, where the temperature is raised to 72 degrees centigrade. Now the application of PCR. Application of PCR in the field of medicine. For the testing of genetic disease mutation, monitoring the gene in gene therapy, detecting disease-causing gene in the parent. Second, the application of PCR in the field of forensic science. It is used as a tool in genetic fingerprinting, identifying the criminal from millions of people, and paternity test. Third, application of PCR in the field of research and genetics. Compare the genome of two organisms in genomic studies. In the phylogenetic analysis of DNA from any source such as fossils. Analysis of gene expression and gene mapping. Now we'll see what are the limitations of PCR. Despite of the widespread application of PCR, this technology has some limitations. PCR cannot be used to amplify unknown targets. Prior information about the target sequence is necessary to design the primers. DNA polymerase error potentially causes mutation in PCR products. थैंक यू फॉर वॉचिंग. अगर आप लोगों को इस वीडियो के हिंदी एक्सप्लेनेशन चाहिए तो प्लीज़ डू